a great honor to have Julio Delgado with us tonight. So Dr. Julio Delgado is a renowned expert in hematological oncology and immunotherapy. He's currently working at Clínic de Barcelona. He has a strong academic background complemented by years of research and clinical practice dedicated to improving outcomes for patients through cutting-edge therapeutic approaches. His expertise spans the entire process of CAR-T development from bench to bedside, making him a leader in this transformative field. So, Julio Delgado, the floor is yours. Thank you very much for being with us tonight.
Okay, thank you very much for your kind introduction. I'm going to share my screen now. I hope you can see my slides. All right.
Not now. Yes, now it's okay.
It's okay now? Okay.
Yes, it's okay.
So thank you very much for your kind invitation. I'm really pleased to be with you today. So that's my name, is Julio Delgado, and I work at Hospital Clínic Barcelona. I'm a hematologist. And before I start, I'd like to show you my conflicts of interest. I don't have any conflict of interest from pharmaceutical companies, but I have to say that I'm employed by Hospital Clínic Barcelona, which is the owner of 3 products that I will mention during my talk. And I was even principal investigator in 2 of these trials. Of course, this is a public academic institution, and it doesn't really matter if we manufacture 10 or 100 products. My salary remains exactly the same, sadly. But I guess you could see this as a conflict of interest, so it's important for you to understand this. So there's no need to talk about CAR T cells. There's no need for introduction. Everybody knows what CAR T cells are. Just to highlight that there are actually 6 products approved in the European Union for the treatment of a variety of hematological malignancies. There's a 7th product that was just approved in the US, but not available, not approved yet in the European Union. The problem, and this slide is a bit old now. It was done in May last year. The problem is that the fact that these products are approved in the European Union does not necessarily mean that these products are available to patients. This is a survey I did in May 2024, and you can see that there's lots and lots of indications that are actually not available to EU patients. So there are countries where everything is red. There's basically no CAR T cell products available. So there's a clear unmet medical need in terms of CAR T cell availability within the European Union. Let's not talk about other jurisdictions. Let's not talk about Asia, Africa, Latin America. So there's a huge problem in having these products delivered to patients. But even if we consider countries like ourselves, like Spain, France, Germany, Italy, you can see from this study that only a minority of patients who are eligible for these products actually get them. And I was going to say that actually France is doing pretty well in this study. 42% of patients who were eligible for CAR T cell therapy for lymphoma actually got the product. But there are countries that are doing far worse, like Italy, where only 17% of patients who are eligible for a commercial CAR T cell product got it in the end. So let's go back a bit in time, and I'm going to show you a paper that changed my life. This paper was published in the summer of 2011, and it changed my life because at the time I was the CLL expert in my department. So that means that when the immunologist who was interested in developing this treatment came to a hematology department, he asked for the CLL guy, and that was me. And that explains why I am the leader and I'm leading these efforts in my institution. And the reason is that the first important paper was on a patient with chronic lymphocytic leukemia. And it all started then. 2 years later, we working together, immunology, hematology, and other colleagues that joined later, we decided to develop our own product. There were several reasons why we could do that. The first one was that we had anti-CD19 monoclonal antibodies available. So we didn't have to worry about intellectual property, about Kymriah, Yescarta. We could create our own thing using our own monoclonal antibody. The second and most important thing was that we had the ability to manufacture our own vectors. This is a crucial bottleneck for academic CAR T cell manufacturing. It's actually the fact that you need vectors, and not everybody can manufacture its own vectors. The third thing was that Clínic's Prodigy came out at the time, so this is a GMP compliant machine that allows you to manufacture CAR T cell products relatively easy. I mean, it's not like a done deal, but it's possible. And then there were other issues like institutional support right from the beginning. The institution showed a lot of support and courage. They encouraged us to carry on doing this. And finally, at the time, even though this was like a conventional CAR T cell product, it was a sort of a replica of what was being developed by Novartis and Yescarta, we reasoned that these products were going to be quite expensive once approved, and having our own product would be very useful for us moving forward. So this is the reason why we decided to develop our own product, even though everybody was telling us that, we're wasting our time, you're simply replicating what pharma is doing. There's no innovation there, go back to the lab, stop thinking like pharma, blah, blah, blah. So we got a lot of negative feedback. I know it's hard to imagine, but that was what we were getting at the time. Anyway, I mean, I was still the CLL guy, and what happened was that that product, Kymriah and others, when tested in clinical trials, the results in CLL were not as good as they were in ALL and non-Hodgkin's lymphoma. And of course, pharma companies, they're interested in trying to get a blockbuster, trying to make money out of their investments. So they decided to ignore CLL and go for non-Hodgkin's lymphoma and ALL. But you see, there are CLL patients out there who need these products. And let's talk about somebody else. Let's talk about the product that was developed at the Hutchinson. That is the product that is actually approved in the US, not in the European Union. This is a product that can be given to these patients. We all have hard to treat CLL patients who would certainly benefit from a CAR T cell product. Well, we have one. So we can offer our product as compassionate use in this instance to patients with CLL, or even worse, patients with Richter's transformation. Richter's transformation, at least in Spain, is outside the approved label for Yescarta or Kymriah. We're not allowed to give commercial products to patients with Richter's transformation. That means we need an alternative, and this alternative could be our own product. So you can see the series. This is unpublished data, but was recently updated a few weeks ago. We've treated so far almost 30 patients with CLL. You see very heavily pretreated, as you would expect from this type of compassionate use program. Complex karyotype, TP53, Richter's transformation, elevated LEH. So you see all the bad prognostic signs for this patient population. The outcome of this is that, well, surprisingly for us, the duration of our own product in these patients with CLL is pretty long, and we don't really understand why, but our product lasts for a long, long time in these patients. And I'm saying surprising because, most of these patients have, immune deficiency. So their T cells should not be as good as in other hematological malignancies, but for some reason, the CAR T cells we manufacture are very durable. Second thing you could notice from the table on the bottom is that the toxicity of our product is not bad at all. Grade 3 or higher CRS, 7%, grade 3 or higher ICANS, 0%. And the reason why is this is because we give our product in fractions. We give 10% of a product if the patient is doing well, we give 30% if the patient is doing well, we give 60% of the product. So we tailor the dose to the patient's outcome and the patient's toxicity. If the patient develops CRS, grade 2 or higher, requiring tocilizumab, etc., we stop cell administration. And this is another advantage of having your own product. You can decide what dose you give, how you give it, etc. And these are the efficacy results. For CLL, 85% of the patients achieve complete response. In Richter's transformation, complete response rate is a bit lower, 69%, with some partial responses. This is a swimmer plot of this patient population, and I'm showing you progression-free of the entire cohort and also divided by CLL only or CLL plus Richter's. And you see that they are basically the same. So even in this hard-to-treat population, patients with Richter's transformation, we achieve long-term disease control with our own product, which is quite encouraging. And we're not the only one. There's a group in Heidelberg, and is my dear colleague Michael Schmidt. They have their own product, also academic, and they are using it to treat, for instance, CLL, because this is a disease where there are no commercial products available, and they have their own results, perhaps not as good as ours, but very interesting indeed. They published their results last year. One thing we've noticed is that particularly in cases with Richter's transformation, there's a risk of CD19 negative relapse. We've seen that in 3 patients. And that worries us a little bit because, you know, even though we try to anticipate and give and refer and recommend CAR T cells to patients who are at high risk but not transformed yet, the reality is that most patients are referred to us when they have already transformed. So that means Richter's transformation is a common patient population that is referred to us. So we've been thinking about, well, you know, if we're having CD19 negative relapses in these patients with Richter's transformation, let's think about an alternative. And you may know we have another product. I'm not going to talk a lot about this one. It's called RE2 or sesnicaptogene. This is an BCMA CAR T cell product. And the results of this trial involving 60 patients were updated last year in this publication, Clinical Cancer Research, median PFS of 16 months in this patient population of triple-class exposed multiple myeloma. But what I'm trying to get to here is that most lymphoma subtypes do express BCMA as well. So BCMA is an antigen that is, of course, expressed in plasma cells. That's why we use it for the treatment of myeloma, but it's also expressed on many lymphoma subtypes. So we said, okay, we have RE1, we have RE2, let's do RE3. Again, another advantage of having your own products. You can play with them, you can try to innovate. And we designed this product that targets both antigens at the same time, CD19 and BCMA. We tried to see what was the best combination. We looked into different approaches. One, the simplest, would be just infusing both products at the same time, co-infusion, but that's the most expensive one because you have to manufacture 2 products for 1 patient. We tested co-transduction, which is culturing cells with both vectors at the same time. And we tested also bicistronic and tandem configurations. But at the end of the day, we found that the best possible combination was co-transduction, and that is what we do. So for our RE3 product, we culture the T cells with both vectors at the same time, and you end up with these cells. Some of them have both constructs, some have only one, some have the other, and this is the product we infuse. This clinical trial opened in June 2024. We've already included 7 patients, I believe. And the target population for this trial are basically patients who are ineligible for commercial CAR T-cell products. For instance, patients with Richter's transformation, patients with Burkitt's lymphoma, patients with aggressive lymphoma that are ineligible for commercial CAR T-cell therapy, or patients who have already received CAR T-cell, Yescarta, Kymriah, whatever, and they have relapsed lymphoma. So this is what we're testing right now, and we hope to have some data soon. I can tell you, for what we've done, that at least safety is equivalent to RE2 and RE1. There's no unexpected adverse events. But I'm going to start being a bit philosophical now, is how to self-sustain your academic program. Because, of course, this is what we normally do. We are scientists, we are doctors, so we have a question: how do we improve the outcome of ALL patients, for instance, or lymphoma patients, or CLL patients? We ask for money, we do complex preclinical studies, functional studies, we design a clinical trial aimed at answering that question, and our goal is to publish our results in the best possible journal. I don't know, New England if you can, or Nature Medicine. You know, the French group, for instance, has been, amazingly productive with their database. They've been able to publish amazing papers in the best possible journals. And perhaps license your idea, if it's a new idea, to a company. But we've become drug developers. We have a different approach. So the questions we have are regulatory. My question is not, how do I improve the outcome of my patients? My question is, how do I get my drug approved? I request funding all the same, but then I contact regulators and I do the minimum preclinical studies needed to start my trial. I'm not interested in functional studies. I'm not interested in complex, expensive, labor-intensive tests that are needed to publish these results in good journals. I'm only interested in the minimum tests I need so that the regulators can accept my product. Then I contact regulators again, and I design my clinical trial aimed at getting the drug approved. I don't care about what is the best possible scientific solution, what would be the most interesting trial. I basically behave as a pharmaceutical company. I simply design a trial that would allow me to go and get a marketing authorization application. And perhaps maybe publish our results. But that is not my primary goal. My primary goal is to get my drug approved, because that's the only way you're going to self-sustain. You cannot manufacture your academic products forever and ever, because if they're not approved, you will struggle to get them reimbursed. And what we are trying is adult acute lymphoblastic leukemia, because that when we started, we thought that was where the unmet medical need was. And this is an update of results from last year. We've now treated more patients. These are 60 adults treated. And you can see from the slide, the complete response, 90%, median duration of response, 13 months, median PFS, 12 months. And again, toxicity is quite reasonable, 4% grade 3 or higher CRS, 1% grade 3 or higher ICANS. Again, the reason because we give this drug in fractions. If you compare that with the product that is currently approved in the European Union, although not available in Spain, I know that it's available in France, but it's not available, it's not reimbursed in Spain, you can see that basically the efficacy is the same, the PFS is the same, the response rate is the same, and perhaps the toxicity is a bit worse. And here I would like to remind you that we are in the European Union, and the European Union has two ways of getting drugs approved. One, the centralized procedure, the one that we should all follow, but there's another one, hospital exemption, that is available to all of us, including you in France. There are many differences between the two, but if you're serious about developing your academic products, consider the possibility of going to hospital exemption, because you could get your product approved in your own country, and then once it's approved, you could ask for reimbursement to your government. And this is what you need to understand, that there's something called the valley of death. This is not for CAR T cells, it's for drug development in general. The valley of death is this situation where you're just spending money all the time on your preclinical, your clinical trial, etc., until you reach a point where the product is approved and you start to sell something, you start to get some reimbursement, and you can cross the valley of death. And the funny thing about the Valley of Death that exists, it's a real place. It's here in the state of California. And I like this idea, because what happens in reality is that pharma companies start from Hollywood, the land of the dreams, and they wish to cross the Valley of Death, but to end it in Las Vegas, because their interest is to hit the jackpot, to earn as much money as they can. We, our public institution, we would like to cross the valley of death as well. We want our products to be self-sustainable, but instead of going to Las Vegas, we would like to go to Yosemite National Park and live in peace with nature. We're not interested in getting money out of it. We're interested in providing products to patients who wouldn't be eligible for commercial products, or those who live in countries where commercial products do simply not exist. Many countries out there. So how do you make it work? Well, you start thinking as a drug developer. What is your goal? To get your product approved. You contact regulators all the time. You design your non-clinical test, your quality control. You design your clinical trial, but following their advice. You read the guidelines. It's foolish to design your own clinical trial based on what you know about the disease if your goal is to get your drug approved. You have to understand how regulators think. You surround yourself with experts in regulatory science, or even as I did, you go to the EMA yourself. When in 2020, I went to the EMA, I spent there 15 months as secondment national expert, so I could actually understand the language, so I could see what they mean when they talk about significant benefit, major therapeutic advantage, comprehensive data, all that vocabulary that we don't have as physicians, but they do have as regulators. You could ask for prime designation, you could ask for orphan designation, or even there's a pilot program that this program started in 2022 of enhanced support to academic and non-for-profit developers, or ATMPs. And you simply contact the Advanced Therapies Office at the EMA, and they'll tell you what to do. And this is the result of that. This is our pivotal trial. So I showed you what I showed you before were the results of our original trial in adult patients with ALL. Now I'm showing you the pivotal data that we'll send to the EMA very soon, well, not very soon actually, in a few months' time, and try to get our product approved in the European Union. We're trying to be the first academic institution to ever have a product approved centrally in the European Union. These are patients we included in these trials, or adult patients with acute lymphoblastic leukemia, age 18 to 70. They all had to have measurable disease by next generation flow, at least, because the primary endpoint was complete response rate at day 28. This is again unpublished data. The efficacy cohort were 32 patients, and you can see here that month 1 at day 28, 85% of patients had complete response with undetectable, measurable residual disease by next generation flow. The per protocol cohort is those patients who receive all the fractions. I told you before that the dose we give to the patient depends on whether they have CRS or ICANS during the cell administration. So only 23 patients were able to receive all the fractions, the entire dose. And in those patients, of course, the response rate was higher. The median duration of response was 12 months in the efficacy cohort, and it was almost, well, 15.6 months in the per protocol cohort, those patients who received the full dose. In terms of safety, we confirm the same. Perhaps the severe CRS was a bit higher in this patient population, 12%. And what we see, probably something you see as well, when you give CAR T cells to patients with acute lymphoblastic leukemia, there's a clear correlation between tumor burden, the blast cell count in the marrow, and the incidence of severe CRS. You see here, all patients who had severe CRS were patients who had a high blast cell count in the marrow. On top of that, we are preparing a confirmatory trial. The European Union, the EMA, said, okay, you're presenting data of patients treated in Spain only, and you're asking for a marketing authorization valid in the European Union. You need to treat patients outside Spain. Luckily, last year we got funding to do that, and we are preparing this trial. This is not easy at all. Simply setting up all manufacturing sites in different places under different jurisdictions, different countries, is costing us a lot of time and effort, but we'll get there. And there'll be French centers, and the funder for French patients is Fondation ARC. And this is how the CAR T cell administration looks like in Hospital Clínic Barcelona. This is to show you that the fact that we have academic products does not mean we are not giving commercial products. So we give Yescarta all the same. So whenever a patient is eligible for Yescarta, we provide them with Yescarta. But of course, we give a lot of Varcel, RE1, a lot of Sesnicel, RE2. We also take part in clinical trials sponsored by Novartis, Janssen, all the key players. So we help develop other products as well. The only difference is that we can provide our own product to patients who have no alternative. And you could say, yeah, okay, this is because you are in Spain. You showed us before there are many products that are not available in your country. So that explains why you can give academic products. Okay, let's go to Germany. Germany is the only EU member state where all CAR T cell products, all of them, are available the following day. They are approved by the European Commission. And there, in Germany, also academic products have a role. They are, this is a slide kindly given to me by Professor Michael Schmidt again. You can see that they've treated less patients. We've treated almost 450 since we started in 2017. They've reached almost the 300 mark, but still it's the same. So their academic products coexist with commercial products. No harm done. We are not blocking any development. We are not jeopardizing anybody's business. We are simply providing solutions to patients. As simple as that. And then, well, once you've done RE1, RE2, RE3, well, you try other things because, you know, well, the fourth product is easier than the first and the fifth is easier than the fourth. So very soon we're going to start trying solid tumors, which is, I am going to tell that to you, much more difficult. And we'll see how it goes. But of course, we're not so sure this is going to be successful. We're going to try HER2 breast cancer. We're going to use a monoclonal antibody that has low affinity because, as you know very well, HER2 is expressed in many normal tissues. So we're not interested in killing all cells expressing HER2, only the cells that have a high HER2 expression, which are malignant cells. So this is the product we are going to test. We got funding last year. We've completed all validations, and we hope to start this clinical trial this summer. This will be the first time we treat patients with solid tumors with a CAR T cell product manufactured and created at our institution. And finally, well, we're reaching the end of my talk. I'd like to tell you how we organize ourselves. And the first thing I'm going to say is that this is a huge team effort, huge team effort. So you're hearing me, but I'm just a pawn. I'm just one member of a team. There are many, many people involved. The three main departments involved are hematology oncology on the one hand, immunology, because they are the ones who manufacture the cells and the vectors, and very importantly, clinical pharmacology, because as you may understand, there's a lot of documents to be prepared. The IMP dossier, the investigator's brochure, all those documents that normally sponsors provide to you when you start a clinical trial, well, these ones you have to write yourself. It's a lot of work. You need a lot of expertise and a lot of people willing to spend their time doing that, preparing documentation so you can start your own clinical trials and hope for an approval, which is the goal really, to get the drugs approved so you can ask for reimbursement and self-sustain your CAR T-cell program. In the middle of it, the institution. If you don't have institutional support, you can forget about this. You need a lot of institutional support because there's a lot of hurdles out there. There's a lot of pharma who hate our guts. There's a lot of people who want us to fail. And you need support from your hospital, from the general director, from the clinical director, the medical director, people who understand how important this is, it is what you're doing. And then, well, other departments, as with any CAR T-cell program, there's no difference here. Neurology, pathology, microbiology, intensive care, IRB, ethics committee, pharmacy, quality, etc. I'm just concluding now to say that patient access to commercial T-cells is challenging, even in wealthy countries. Academic products provide a solution in situations where there are little or no alternatives. Having your own products allows you to improve them, alter the dose, the administration mode, etc. Commercial and academic products complement each other. That's important. Availability of academic products does not impair access to commercial products. Academic developments are possible. Trust me, they are possible, but with the appropriate mindset. Not with a scientific mindset, but with a drug developer mindset. And sometimes it feels like you are like Sisyphus trying to push the rock up the mountain. Sometimes the rock goes down again. It's hard. You need stamina. You need, to be positive about it. But I'm very sure we'll make it to the top. And like Sisyphus, we'll make it to the top of the mountain with our own products. Just a sample of the people helping me, just a small, really, there are many more. But here are in bold type people who recruited patients or treated patients with CLL and ALL, the unpublished data I showed to you. Valentin, Neil, and Nuria, they're the ones who collected the data as well. They're the ones running the CAR T-cell clinic, and then other various people. And these are the funders that are supporting us and anybody else who wish to contribute. we can make the logos smaller. It's okay. We can put more. No problem with that. And thank you very much.
Thank you. Thank you very much for the nice presentation. So we will now move on to the Q&A session. So please, everyone, do not hesitate to ask your question on the Q&A part of the. Maybe I can start with one. I was wondering if you have tested one of your anti-CD19 product on autoimmune diseases.
Yes. Well, we have, but you see, this is something that is a bit difficult for us because our colleagues from autoimmune disease department, they are not very keen on that. So at the end of the day, you need the physician in charge to send you the patients. But we've done that with some autoimmune diseases. We are trying to get funding for a proper trial as well. But so far, this has not been like one of our goals. But we think RE1 could be useful. We also think RE3 could be useful because you see with RE3, you also get rid of plasma cells, not only because it's BCMA and CD19. So we think that with RE3, we would achieve a deeper eradication of antibodies, autoantibodies, and perhaps they would be even more effective. And actually, we are trying to get funding also for other various projects in not even not just autoimmune diseases, but even prevention of renal graft rejection, prevention of liver graft rejection, autoimmune hepatitis. Rare autoimmune diseases and other situations because lupus, I mean, there are products already out there and we're a bit late for lupus. So we're trying other situation perhaps. Also, I can't do everything. So we have a lot of work already. We're preparing a marketing authorization application. So unless somebody from the autoimmune diseases department is willing to actually take the time and do it and write and design, of course I'll help him, but I can't do it, myself. I'm not an expert and it's just a lot of work. Every development takes a lot of time. But yeah, we've tried a couple of patients. We're gonna treat one patient. One patient came to us today actually with systemic sclerosis, patient from actually a different region in Spain, and she came from Madrid, and she'll have RE1 as compassionate use.
We have some question in the chat, and maybe I will start with one, maybe general. I read the question of Bertrand Nadel. For those who want to jump and invest, could you let us know whether you manage to be financially balanced or if public funding needs to be massively invested for a while? And it was one of my questions. How do you manage the financial part between your hospital, the academic national investor?
Yeah, okay. So the first thing is that investors per se, they're not investors because when you invest, normally you're seeking some return. And when you invest in our public hospital, you get no return whatsoever. So it's more like a donation. So we received some donations when we started. It was a lot of crowdfunding when we started, but nowadays we have the first product approved for the treatment of adult ALL in Spain under hospital exemption. So that means the product is approved. Same sort of approval you would get for Yescarta. The difference, of course, is the price. The price of our product is 89,000 because we're non-for-profit. So we go to the Ministry of Health and say, hey, this is what it costs to manufacture the product, and this is what we get. So for that indication, it's approved and reimbursed. So that means the Ministry of Health gives you back the money if you send the patient for that. The second product, RE2, was approved on the hospital exemption as well for the treatment of multiple myeloma. Again, there were no products reimbursed in Spain at the time. And now we're trying to get it reimbursed, negotiating as we were a pharma company. So for RE2 in myeloma, eventually we hope it will be reimbursed as well. So same as when you prescribe ibrutinib to a patient with CLL. You pay for it and then you ask your government for reimbursement and you get the money back because it's approved for that indication. So it's exactly the same mechanism. For compassionate use, though, it's different because the product for CLL, the product is not approved. So that means the regional health authority sending the patient to us has to pay 89,000. That's what it is. It's not for free. So if a patient from Madrid, from Galicia, from Andalusia is referred to us for CAR T cells as compassionate use, we charge them 89,000. And well, some regional authorities are willing to pay, some are not, and then, well, we get the patients or we don't. We do have a fixed money for our own patients in Catalonia. So Catalonia, because healthcare is regional in Spain. So actually the government that is actually paying for drugs, etc., is not the central government, it's the regional government. So we have a fixed amount for our own patients. And that was sent a few years ago, and the Catalonian government sees the value in this program, and also because all the patients we are attracting from other regions and how we're growing, and of course, you know, the institution is growing. So they see the value in our institution having this program out there. So that's why they're funding it. But at the end of the day, you need to get your drugs approved. So that is why an academic institution, even an academic institution that is developing the products, cannot simply say, okay, I'm going to do a trial and then another one and then another one, and then ask for more money, another grant, another trial. That is not sustainable. You need to start thinking as a pharma company. You need to start thinking as, okay, my goal is not simply to do a trial. It's not simply to publish a paper. My goal is to get the drug approved. And if it's approved, then it'll be reimbursed. And that is what we're trying now. So hopefully, if everything goes according to plan, we may get the drug approved in the European Union. If that's the case, for instance, in France, let's talk about France. So our partners in France are UFS, and first facility will be in Besançon, and they will be our, let's say, partner. And if the product is approved, then they will go to the French Ministry of Health with us because we own the product, but they'll be the manufacturers of our product, and we will negotiate the price with the French government. If the French government says, well, we're not interested, well, okay, but for you, you won't get it, as with any other drug. If the French government is interested, then it'll be available for French patients in that particular indication. Same as any other product. The only difference being that the manufacturer will be national. We cannot manufacture CAR T-cell products for the entire European Union. Besides the fact that exporting ATMPs is quite complex and not really possible for a public institution. So in order to export, you need to be a private company, as EU legislation currently stands. So we need help from a network of academic public institutions willing to do the same thing, which is to manufacture these products without a profit, simply to provide these products to patients, getting their money back, of course, but without a profit in mind.
Thank you. We have many other questions. So one from Alexis Talbot. He's wondering, how will RE5, 6, and so on look like? Do you have any plan to use CRISPR-Cas9, for example?
Well, yes, we are trying um, other, yes. I mean, of course, we are trying with dual constructs for T cell malignancies. We're testing one for AML, uh, but at the same time, doing the preclinical thing is one thing, and then making it till the end, doing the manufacturing, finding money for the trial is a very different thing. It's a different scale completely. So I'm not sure we will be able to make it to the patients or to get them approved, all of them. We may need help at some point. We may need to license some of them to companies. I mean, I don't, unless we grow, I mean, exponentially, and then, the entire hospital devotes itself to this, I don't see how we could actually work as a pharmaceutical company. So yes, we are doing stuff. I cannot tell you a lot about because, of course, there's some issues that can't be discussed. But yes, I mean, there's of course the preclinical part of our CAR T-cell program is also growing a lot.
Okay. I may switch to the second question, maybe technical, with the question of Loïc Isbert in Toulouse. What type of CD19 CFV did you use? And do you know the affinity enough rate of the ligand? I am asking because 4-1BB based CAR expressing modified CFV showed superior proliferation and response in preclinical settings, like the impressive expansion you observed in CLL. Maybe you can
Well,
comment.
first of all, I'd like to thank Loic. I mean, I know him. He knows me from the time I was a CLL guy. You see, we were both CLL guys at the time. I'm not anymore. Well, I'm still. I mean, I have my clinic and my CLL patients, but I do other stuff right now. So thank you, Loic, for that question. So I could give you, a very scientific answer to that question, but I'm going to be fully honest with you. We chose the monoclonal we had, and we didn't do a lot of testing. We saw that in vitro it behaved quite similar actually to FMC63, which is the SCFV that all commercial CAR T cells use. So there was not a lot of difference. And that's actually what we needed to see because, we wanted to, our goal was not to innovate. Our goal was not to differentiate ourselves. Our goal was to actually replicate because we wanted to be sure that what we were doing was going to be at least as effective as commercial products. So we had actually the opposite view. So normally you would say, okay, let's try and do something different, something innovative. No, we were doing quite the opposite. Let's try to replicate the same and at the same time try to avoid intellectual property issues. So let's substitute the SCFV by one that was developed by an immunologist. That's a funny story because this anti-CD19 monoclonal antibody was developed in the 90s and was there in a drawer and nobody was using it. And we took it and put it in our construct and checked that it was actually working. Now that we are sending our dossier to the EMA, we need to demonstrate that our product is non-similar compared to commercial CAR T cells as a requirement. If you're not able to demonstrate non-similarity, you cannot get your drug approved because that's considered to be a replica. So we're seeing that now, that actually the affinity is different. So there are differences there that explain, for instance, why we have such a low ICANS rate, incredibly low ICANS rate. And that was not intended. So that happened by chance.
Maybe just before the next question, Marion, if you allow me, I want to just emphasize on the importance for the institution or the consortium to have the this type of opportunity, which means patent and discovery of anti-CD19 that nobody used during 10 years, but it's here and it's yours, and you can invest on this resource. And we know that it's quite hard to patent new discovery and to keep it in the hand of the institution. So can you comment maybe briefly on this and the politic of the institution about this type of discovery?
Yeah. I think I emphasized that, and I would like to overemphasize that again. So you cannot do this on your own. So you need a lot of support, because there are times when you need to convince politicians, when you need to talk to, like very important people. And if you as an investigator, Julio Delgado, going to talk to the minister or, whatever, I mean, you go nowhere. You don't get to talk to that person. But the general director of your hospital could eventually talk to those people. So you need him or her on board from day one. So you need to go and talk to them and talk, and you need that. And he or she will then talk to the legal department and say, hey, you have to help that guy because, there's this CAR T-cell product, blah, blah. So then the lawyers are with you as well, or even, as it's the case, because we are handling so many contracts nowadays, that's been outsourced. The hospital is paying outside lawyers to help the ATMP program, the CAR T-cell program, because we have so many agreements with academic institutions who want to do the same thing, blah, 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 that we need a lot of help. So absolutely, that is very important. And patent, it's a funny thing, but we weren't that interested in intellectual property because we said, we're not for profit. We don't care. What if you want to use our product? Come on, use it. So when we started, we never thought that was important for us. And because that's expensive. It's expensive to pay for a patent, to pay for the lawyers, to do the studies, etc. But then we realized something that is quite funny, that you cannot ask for a name of your product if you don't have intellectual property. You cannot go to the WHO and ask for an INN, an international non-proprietary name, unless you have a patent. So we actually were forced to do that and contract, lawyers, blah, 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 because that was the only way we could get the drug approved. If you don't have IP, you cannot get it approved because you can't even get a name. So it's quite interesting, this. But when we started, we weren't that concerned at all. And the other thing is, when we started, many people were saying, oh, Novartis is going to say this and Gilead, and they're going to sue you, or they're going to block your development. Wow, you hear all that negative feedback. And we could have stopped and say, yeah, you're right. Yeah, they could stop it because, well, it's second generation. They could say there's a patent out there. But we forgot about that. We just simply carried on because imagine that this happens. Imagine that tomorrow Novartis decides to sue us or Gilead. We've already treated hundreds of patients and nobody is going to take that away from us. So even if we fail, even if we cannot make it to the EMA, even if they bankrupt the system, whatever, I've heard things like Novartis is going to sue Spain, the country, for granting hospital exemption to an academic CAR T-cell product. I've heard things like that. So if they do that, if they eventually do that, okay. Well, I mean, it's not going to look nice on you blocking a non-for-profit academic institution. But even if they do that and they succeed, the patients are already treated. Nobody will take that away from us. So that's why we carry on. We ignore all this negative feedback. We ignore people saying, how are you going to do that? You're academic, you will never be able to distribute, you will never be able, all that sort of negative feedback. We simply ignore all that because in the process, we are treating patients and patients and patients, patients who did not have any other option, patients for whom their alternative was simply wait, to die. So carry on, no fear. Don't worry about IP. Don't worry about negative feedback. Don't worry about pharma. If you truly believe in doing this, do it. No excuses.
I think we will carry on, Marion.
Yeah, there are two related questions, so I'm going to ask the two at the same time. The first one was how easy it was to set up the placement at EMA. And the related question is, which were the biggest regulatory challenges you encountered, acceptance by authorities, other, and if you had to do it again, would you do things differently and how?
So going to the EMA, there's actually calls for seconded national experts positions. And so they are, it's not all the time. You have to go to the EMA's website and see if there are, so they are very interested in experts going there because they get a lot of information from you. You can provide them with advice, direct advice on, myeloma, leukemia. Whenever there's a procedure, they ask you stuff and they can ask you immediately. It's quite advantageous for them as well. And if you are interested in drug regulation, are you interested, you're developing your product, is very, very important. So the other thing, it's that the EMA pays really well. And not only you, but your institution. So during the time you are away, they pay your institution for your own salary so they can hire somebody to do your job. And they pay you as well because you keep receiving your own salary plus a supplement from them. So it's actually interesting as well from that point of view. So of course there needs to be a call and you need to apply for that, but it's not that difficult because how many people do you know who've tried that? So it's not that many people who knows this is possible or this is useful or interesting. So there's not a lot of competition. I'm gonna tell you an anecdote now. When I decided that in 2020, I told my boss, and my boss was puzzled, and he was like, "What? Why on earth you want to go to the EMA? What's the point? I mean, go to Stanford, go to Memorial, the usual thing, go to Seattle." And I said, "Well, what to do? Learn what? RNA-seq? No. What I need to learn is not that. I need to learn the language regulators use." So for me, it was very, very important. But not everybody understands this. Maybe your own boss wouldn't understand it if you proposed that to him, as it happened to me. Then when I came back, he said, "Oh, that was a very good idea you had going there." Okay. But that was after the fact. But it's not only me. I mean, I have a team. I mentioned the Department of Clinical Pharmacology. This department has grown a lot. It was a small department. Now it's a lot of people working there. There are like at least 3 or 4 people working on ATMPs only, on advanced therapies only, because of all these products I've mentioned, so all these dossiers, etc. So we have a lot of support from people in the hospital who understand regulation as well. So it was not just me, the freaky guy, the freaky hematologist who went to the EMA, but also people whose job is actually to understand drug regulation and drug development. So that is very important too. And I don't know what I would have done differently, but perhaps I would have read the guidelines before, because there are many things I did wrong when I started because I didn't know. I mean, I was when you design your first trial, etc. So many things I learned later. So what is the appropriate endpoint with the single arm trial? Was the appropriate endpoint for a randomized trial? That sort of stuff we don't know as clinicians, and you understand when you go there, and you don't make mistakes when you talk to regulators, when you understand all that. So these are things that we had to fix later. But besides that, I think we're pretty well set.
Thank you.
Maybe we can make short answers because there is a
All
lot
right.
for the
Okay.
next one, but I have a naive question, but I think it's important to clarify. Thank you for this interesting talk. It's Celia Orlando. We work in our hospital. This may be a naive question, but what criteria do you use to decide whether to administer clinical CAR-T and your own academic ones? I understand that it's only for patients ineligible to CAR-T cell, but I think it's important to pass 1 minute to clarify this point for the audience.
Yes. I mean, some people who don't like us tend to say that we are neglecting patients, that we are not providing patients with approved products because we tend to use our product instead. And that is not true. That is not true. We do sometimes offer our patients clinical trials with our own products, but that is, you know what a clinical trial is. You tell the patient you could have an approved product or you could try this product. That happens all the time. But for the compassionate use program, we never offer compassionate use to a patient who could receive an approved product that is available in our country. Because again, there's a difference between a product being approved and a product being available. And on top of that, there are products that being available and approved, perhaps the waiting list is very long. Some products, multiple myeloma products. Well, in those situations, the physician may approach us and say, would you be able to manufacture this product for our patient in less time? Because, rapidly progressive disease. And we say yes or no, and we offer that to the patient. But we always say, why is this patient not eligible for that commercial product? Okay? Always. Some people don't believe me when I say this, but it's the truth. You see, my salary remains the same. I don't get, I wish it was better. I mean, if there are, 100 academic products administered, my salary goes high, but no, it's exactly the same. Exactly the same. No advantage for me. It's more work.
Thank you. Maybe one other question from Bertrand Adel. Are you entering the race of in vivo CAR-T? Safety is not de-risked.
No.
No?
It's too much. No, I'm sorry. That is no. Ooh.
Because the
No.
costs should be less.
Yes. Oh yeah, absolutely. No. I'm not saying it's not interesting. I'm simply saying we cannot do everything. But that requires, but of course, it may be the future. But we'll see. Because allogeneic products, some people said a few years ago, yeah, allogeneic products are the future. This has not been proven yet. But we'll see. Yeah, it may well happen that the products we're manufacturing now are no longer useful in the future, yes. But for the time being, they are very useful. But no, it's just too much. No. I wish I could. I mean, I wish I had hundreds of people working in the CAR T-cell program, but it's not the case.
Maybe a different question, but I think it's important. Professor McIntyre asked if you plan to use the hospital exemption track long term or hand over to an industrial provider at some stage under appropriate conditions. How do you see the future for this procedure for your product?
Yeah, hospital exemption, we see it as a bridge. So we never see hospital exemption as the end stage of our development. So we ask for hospital exemption for RE1 and then again for RE2 because of what I said before, because once it's approved, it's possibly reimbursed, and you need some money back during your development. And EMA, it's a different story. It's much more complex, a lot more money needed to prepare all the documents, to prepare all the things you have to do, pediatric investigation plan, non-similarity. There's a lot of stuff there. So for us, hospital exemption is a bridge. It's something we use in the meantime. And as an example, RE1, the hospital exemption approval is adult ALL, and the approval we're seeking in the European Union is adult ALL. It's exactly the same. So we follow like this line here. And for multiple myeloma, exactly the same. So it is approved for triple classes plus blah, blah. And now we are designing a trial in Europe, because the EMA told us that that one needs to be done in Europe directly, which is not easy to get funding, by the way, for an academic trial, pivotal trial. That's many millions, and there's no horizon call for that. And we're designing that trial, and hopefully we'll get it done, so we can extend that to the entire European Union. So from hospital exemption in Spain to EMA. But well, not easy. I mean, not easy, and I'm not saying we're going to succeed, because I don't know. We may not. We may not. We will try. We will certainly try. We may be the first to succeed or the first to fail. We'll see. But even if we fail, you know, we will sort of demonstrate the regulators that this is possible, that academia can do stuff, that we're at least as bright as pharma people. We can do the same thing. Why not? Why are some people saying you are academia, you cannot, you will never be like pharma? Why not? Why some people think that pharma people are brighter? They're not. We are at least as bright as them.
But you mentioned important things, that you have to think as a drug developer. You have to accept the implication of quality process and
legal responsibility,
all the checkpoints. To accept to spend a lot of time on these things because it's important.
Yes. If you wanna do like pharma, you have to behave like pharma. Yes, absolutely. But even if we fail, I mean, somebody will succeed. Maybe the second one or the third one, I don't know, but we'll see with our own eyes an academic product being approved by the EMA. Trust me on this one. We'll see one. If not ours, somebody else's. But this will happen. Why? Because commercial CAR T-cell products are not sustainable. Commercial gene therapies are not sustainable. Not in the US, not in the EU, nowhere. I'm sorry. They are not sustainable. So we'll need to find a solution. And this is the one we believe is the best one. We doing it ourselves.
We are maybe going to take a last question, which will be the one a bit provocative. Would you be interested by an indirect comparison between an academic CAR T and, your academic CAR T and commercial CAR T therapy?
It's not a question of being interested, is that we need to do it. We are going to the EMA with a single arm trial. And that means we will need an indirect comparison, and we are already collecting data to do that. It's an absolute requirement. Unless you have randomized data, which we don't, you need an indirect comparison and a lot more things. I'm not going to go into the details, but we've already met with the EMA and discussed everything we need for this marketing authorization application. And one of the things is that. If we don't get enough data in Spain, because of course all the patients have been treated in Spain, and we thought that the best comparator should come from Spain too. we may go to other countries. But this indirect comparison does not need to be with other CAR T cells. Does not need to be with other CAR T cells. It can be with approved products in that indication, that is relapsed refractory acute lymphoblastic leukemia in adults. That's what pharma does, exactly that. So we don't need to reinvent the wheel. We don't need to do something different simply because the paper is going to be nicer. We need to do what it's needed to do, what we need to get the drug approved. So we will do an indirect comparison. Yes, we must. That's a requirement.
Thank you. So thank you very much for the presentation, Julio, and thank you to all of you for your participation and the discussion, the questions. Very interesting.
Thank you. Thank you for having me. It was great.
So yes, thank you all and see you soon for the next Red Talks.